Eddie and I tested for coliform bacteria in the Oakford lake water. The importance of chloroform bacteria. It is used to indicate the quality of food and water. It is found in large numbers in feces of warm-blooded animals, and coliforms are not normally the cause of serious illness. Their presence indicates that other pathogenic or disease-causing organisms are present. Why should we care about coliform bacteria? If coliform bacteria is present, that indicates that there could be harmful organisms or fecal matter within the lake. Fecal matter can be classified as either human or animal waste. This graph shows the acceptable levels of fecal coliform bacteria in water. The materials you'll need to test for chloroform bacteria in the lake water consist of an incubator, 2 different lake samples, lactose broth with chlorophenol red, you need 12 of them. You'll need different pipettes and tap and distilled water to use as the control. All of these vials contain lactose broth chlorophenol to use as an indicator. The D and T labeled vials stand for distilled water and tap water, while 5 of the vials are labeled number 2, meaning that they came from a different source than the other 5 vials labeled number 1. The vial with the green cap on it represents the water from the lake. The vial labeled D stands for distilled water, and this picture is taken before the incubation process. The vial labeled T stands for tap water, and this is also taken before the incubation process. In our experiment, we tested the tap water, distilled water, and water from 2 places on the lake. All of the samples, except for the distilled water, had coliform density of 100 that was greater than 180 over 100 milliliters. We went through some problems going through this experiment because of the temperature in the incubator fluctuated between 38 and 37 degrees Celsius, and it never stayed at a constant temperature. The test was simplified so it could only test positive or negative without any levels. The procedure. First, put 1 milliliter of one lake sample into 5 of the lactose broth vials. Then put another milliliter of the other lake sample into the other 5 lactose broth vials. Put 1 milliliter of tap water into one of the unfilled lactose broth vials. And finally, put 1 milliliter of distilled water into the last lactose broth vial. Incubate all of the samples for 48 hours at a constant temperature. In this case, we used 35 degrees Celsius. After the 48 hours is completed, remove the samples from the incubator and observe changes in color. If you see a change in color, it will indicate that there was chloroform bacteria within the water.